Hello, my name is Caroline Kwon, and I had the privilege of working at the Davis Lab at the VA hospital this summer. And today I'm just going to be presenting a little bit about the research I was able to do about the mechanisms underlying Parkinson's disease and the genetic risk factor GBA. So just as a bit of background on Parkinson's, it's the second most common neurodegenerative disease after Alzheimer's. It affects 1 to 2% of the population over 65. Some common symptoms include tremors, postural instability, rigidity, cognitive decay, stuff like that. And it's also characterized by the presence of Lewy bodies, which are these abnormal aggregates of including ubiquitinated proteins and also this specific protein called alpha-synuclein, which I'll get into. And interestingly, majority of Parkinson cases are actually sporadic, which have no clear genetic or environmental cause. So the spectrum of genetic influences on Parkinson's is actually very complex, and it can range from these rare single causative genes with almost 100% penetrance to very common but clinically insignificant genes. And the gene we look at, GBA, is very interesting in that it's sort of right in the middle. It has clinically significant penetrance. It increases the risk of developing Parkinson's by about 5-fold, and it's present in 5 to 10% of the population. GBA encodes a lysosomal enzyme, and it's known that if you have a recessive mutation in GBA, you actually, it causes Gaucher's disease, which is the most common lysosomal storage disease. But heterozygous GBA mutations are the strongest genetic risk factor for Parkinson's. Like I said, it increases by 5-fold. And GBA carriers with Parkinson's have more rapid disease progression than Parkinson's patients who are non-carriers. So normally when proteins are targeted for lysosome degradation, they're tagged with ubiquitin. In the case of a GBA deficiency, these ubiquitinated proteins sort of aggregate and are unable to be degraded, leading to Lewy pathology. As we can see by the scary red bar, when a fruit fly has a GBA deficiency, the levels of ubiquitin are very, very high. And we can also see, as indicated in the green, that when we reintroduce the GBA into those fruit flies, there's actually a rescue of that aggregation. Previous work in the lab also shows us that when we introduce GBA into a specific tissue that is distant from the brain, there's actually a rescue of aggregation in the brain. And there's a very dramatic decrease, as you can see in these whole brain images. And also, we find that this rescue is mediated by alterations in the vesicular trafficking pathway, and specifically by alterations in extracellular vesicles. So this leads us into our current model and hypothesis, which is that extracellular vesicles are a vehicle for propagating protein aggregation from cell to cell, and that GBA accelerates this process. And we wanted to study the vesicular trafficking important for lysosomal degradation and extracellular vesicle formation to see if those pathways were altered by GBA. So the first model we use in our lab is actually the Drosophila fruit fly GBA loss of function model. So in these flies, we delete the Drosophila homologue of human GBA, which is 50% similar and is expressed ubiquitously. And in these flies, we express human alpha-synuclein, the protein I said was in Lewy bodies, in the muscle and actually the neuronal tissues. And we age them a specific amount of time. It's sort of a long process. And then we separate the neuronal and muscle tissue and run them on a Western blot. We use actin as our control and synuclein as our marker of interest. Synuclein is a very small protein. So GBA deletion causes these proteins to aggregate. And so we see when there is aggregation, these higher molecular weight synuclein oligomers, as we'll see on the gel. And we look for also the low molecular weight synuclein, which is indicative of when there are the synuclein monomers. So yeah, this is the Western I ran actually just last week. On the left, we have the heads, and on the right, we have the thoraces. Unfortunately for this gel, the actin control bands did not actually show up very well, so it was difficult for us to normalize the signal we received from the synuclein. We also saw much less of the high molecular weight synuclein than we expected, especially in the GBA deficient flies, and much more of the low molecular weight synuclein. So future steps for this project would just be to rerun the gel, hopefully with better control lanes and just to be able to see whether there actually is a difference. The second model we use in our lab is the human neuronal GBA Parkinson's model. For this model, we collected fibroblasts from a Parkinson's patient carrying a mutation in GBA, and we produced induced pluripotent stem cells, or iPSCs, from them. We also used those same cells to produce wild-type revertant cells as a control, and collected from a different individual age and sex-matched healthy control cells. So what we did with these cells is we differentiated them into neurons, also a very long and complicated process. We fixed and stained them with different lysosomal markers, each corresponding to a different stage of the vesicular trafficking pathway, and we imaged them using confocal microscopy. So the cool thing about confocal microscopy is that you can focus light through a pinhole to focus on a specific Z plane. So these are what are known as Z stacks, which are a bunch of those planes stacked on top of each other. For these specific cells, on the left we have wild type, on the right we have mutant, and they are marked in green with the lysosomal marker EEA1, which marks the early endosomes. And for each of the 5 lysosomal markers, we imaged 15 neurons per cell line for a total of 150. So these are just 2 of the images I took from, all of those cells. And I think it's really interesting that you can see on the wild type cell, there seems to be a stronger green signal. And I think that is sort of the overall trend I noticed while I was taking those images. But the next step for this project would be to do a normalized quantification of the relative signal strengths in order to be able to make a more definitive statement. All right, I just would like to take this time to acknowledge everyone that made this program possible. Thank you to Dr. Ellenbogen, Dr. McDonald, Dr. Davis, my PI, and Sylvia and Julie for everything you've done. This was a really great summer, so thank you.